All right, everyone. I think it's time to get going here. Welcome to the Emergent Science Committee on using metabolomics for characterizing the all-important human exposome. So this is a part of the Emergent Science Committee from the National Academy of Sciences, sponsored by the National Institute of Environmental Health Sciences, as you know. It's a forum between scientists, decision makers, and investigators to determine the next waves in research and science today. So just to remind everyone, the NRC is going to write a meeting summary. It's not a consensus report, so this committee that convened today will write a summary and not a report that generated from these things that we discussed today. And we also want to, of course, thank you all for coming and welcome all the participants who are attending remotely as well. So welcome to all you folks in Webland coming to us over the internet. We will have QA capabilities, so stay tuned for that. I think that will be pushed your way shortly. So please send in your questions. We'll go over all of those towards the end of this session here. So again, welcome. I first want to thank all of the participants who we invited on behalf of the committee, Marilee Shelton Davenport, Keegan Sawyer, our chair of our Fearless Committee, Dr. Farland, and all of you for participating. So I think we are at a critical juncture now, and we really need to put the pedal to the metal in thinking about how to use these new genomics technologies to ascertain the human exposome. And as you could figure from the title and the roster that we've assembled here, we really think that metabolomics is one way forward of assessing this elusive but emerging data modality. So when we were assembling this roster and also the theme behind this workshop today, we claim that metabolomics technologies can enable the comprehensive and accessible, so accessible meaning easy to use by investigators and cheap, of the high throughput human exposome. So actually upgrading the exposome to the level of the genome today. And that these technologies can accelerate data-driven discovery in health and disease, so it's something that has also been elusive in environmental health sciences, and also have wide-reaching implications in determining health policy and decision-making. So all of this is what we think would be the future for this data modality, and how metabolomics can fit in is what we'll be discussing today. So just to give you some motivation, I'd like to start out with how I started out with teaching our computational genomics folks, and that it's all important to remember that phenotypes are a function of genes plus environment. So when we talk about the phenome, we think about height, your eye color, and also diseases like type 2 diabetes and cancer. And this arises as a function of the genome, so the places where you and I differ, so the 10 million single nucleotide polymorphisms, other structural variants, and so forth. But also the environment, or this concept of the exposome, which can encapsulate this entire equation here. Things such as infectious agents, nutrients, pollutants, pharmaceutical drugs, and on and on and on. To define what means the exposome is at large, and something that we need, also a topic today. But this is the equation in which we want to work with. So phenotypes emerge due to genes and environment. We're exposed to many different factors of the exposome, but we lack methods to ascertain the high throughput exposome. And that's the whole, one of the missions of today's, or one of the visions of today. So in the genome today, we have a unified and accessible, cost-effective platform that has allowed high-throughput discovery of genes and gene variants in disease. And these things are accessible, so they only cost on the order of $100 today, so you could do large populations to ascertain differences along the genome for different diseases of choice. So this has led to 1,400 genome-wide association studies in which we have pretty robust evidence of specific variants that are connected to phenotypes of interest. But of course, this leaves out the other end of the spectrum here, and it's debatable on whether these genomic variants that are found in GWAS are actually clinically actionable. So we need these technologies, these accessible technologies, to ascertain the exposome. So again, back to Genetics 101, let's motivate this a little bit more with some data. So variance in phenotype is equal to variance in genes plus environment. And so human geneticists have a great way of quantifying the effect of things passed from mom and dad in terms of phenotype. So it's this notion of heritability. So heritability is this range of phenotypic variability attributed to things that mom and dad passed down to as genetic variability in the population. And it's simply this ratio of the sigma G over the sigma P. And it can give us a raw indicator of the proportion of phenotypic differences that are attributed to genomic differences. And so you could just download studies, family-based studies, in which they take twins or related individuals to ascertain heritability. And what you'll see is that while GWAS have been quite successful in finding specific variants, we're missing a lot of the variability for burdensome diseases today. So I just took down data from SNPpedia.com in which they ascertain heritability here on the x-axis. And you have everything sorted from 0, so not genetically controlled, all the way to 100. So things that are 100% genetically controlled, as you might expect, include hair curliness or eye color. But all the way at the top are things such as stomach cancer, leukemia, lung cancer, all of which you could probably name the risk factors off the top of your head. But things like type 2 diabetes, longevity, Parkinson's disease, heart hypertension, all have heritabilities less than 20%, which leaves a lot of room to describe this variability with the exposome. So there's a massive opportunity. And not to belabor this point, there is this meta-analysis published by our human genomics friends, Peter Vischer and Tinka Polderman, Nature Genetics. They said, okay, let's scale up all these heritability estimates from the literature. So they looked at 18,000 traits of the phenome. They ascertained about 3,000 publications, looked at over 14 million twin pairs to assess heritability, and they came up with an average heritability across all of these traits as 0.49. So in the end, the exposome plays an equal role, and we need technologies to ascertain the exposome to get to that other 50%. So as you will hear from talks today, the explaining the other 51%, we need a new data-driven and a cost-effective paradigm in which we've begged the metabolomics investigators to help us get there. A data-driven, accessible view of the environment is required to discover the cause of burdensome diseases today. And so as a layer of background, I'll lean on David Balshaw, who comes up next to talk a little bit about this. But we had this exposome workshop in January to kind of define where these topics may go, and to characterize and to define the exposome. So things that we discussed, or we divided up into themes. For example, characterizing what we call the internal exposome, the external exposome, so exposome and how we ascertain biological responses, or these phenotypes, how we introduce the exposome into epidemiological studies and design of epidemiological studies to characterize association and causality. And of course, there's a large data challenge, so we had an exposome data analytics and informatics part that I was proud to be a part of. So just to give you a view of what this might mean, so one of the questions that came out of this is, can metabolomics provide this analogous, unified, and cost-effective data modality for ascertainment of the exposome? So similar to that Illumina chip that I showed you earlier. But there, of course, are many challenges in using this, and this is obviously just this one slide won't do it justice, and what we'll talk about today is that the metabolome is, first of all, both P and E simultaneously. So it'll be very difficult to deconvolve this relationship that metabolome is also a great measurement of phenotype, but also can characterize perhaps E as well. And we also know that the metabolome can be mitigated by genetic factors. So we have GWAS on metabolomic factors too, so we know that there's a correlation there. And some of the other topics that, for example, folks like Toby, Athersuch, and so forth will get to are things to consider, including endogenous versus exogenous exposures, the technologies that we use to ascertain untargeted versus targeted, and study designs and temporality to consider the exposome. But I think there are great possibilities for discovery, and so, for example, we've put together a paper that came out as a result of this exposome workshop, and it's great to imagine these new possibilities that their exposome modality might be. So you can imagine this exposome, it's a data matrix of many different things on the columns, for example, that ascertain the exposome, like pollutants, diet, xenobiotics, drugs, to the gut flora, and you can ascertain this presumably if it's accessible in a longitudinal manner, so that the first time ever we can ascertain mixtures of exposures through time, and this is very important to see how things are correlated together and how they may interact on the exposome level. But then have considered the phenome also longitudinally, so phenomic traits like cardiovascular disease, blood pressure, cancer, weight, height, gene expression, and do these what we'll call exposome-wide or environment-wide association studies to connect the exposome with phenotypes to get to causality, or even do things like phenome-wide association studies to see how exposures are connected to a bag of or a mixture of phenotypes together. So a lot of room for impactful discovery if this data modality is in place and if we can measure them in an accessible way in many individuals over time. So again, that's to set the stage here. I thank you. I get to remind you all, we welcome all our webcast participants. The QA capabilities will be in force, and so so send in your questions. Be sure to send in your questions, and without further ado, I send it on to our next speaker here, David Balshaw.